Hi everyone, welcome to the course Introduction to Protein Engineering 101. In this video, we're going to dive into the first lecture of Module 1, which is how scientists learn to program proteins. Right, so in this lecture, we're going to dive into the concept of undruggable proteins versus druggable proteins. Undruggable proteins actually consist 85% of all human proteins, and they're very important to this idea of drug discovery and drug therapeutics, so it's something that's essential to protein engineering. Just an overview of what we're going to be talking about in this lecture. We're going to look into traditional drug discovery methods, active sites of enzymes, lock and key model, as well as the occupancy-driven pharmacology model, which is a more traditional model of drug discovery, and why most proteins lack binding pockets, as well as a general comparison between undruggable and druggable proteomes. Right, so we're going to start with a very basic question. Why can't we just make a drug for every single disease that's out there? Well, I think to tackle this question, we really need to look at the composition of the human proteome. The human genome contains approximately 30,000 protein coding genes, and proteins execute nearly all of the cellular functions in the body. And diseases often arise precisely because these proteins are dysfunctional. The drug discovery problem that we're encountering in today's medicine is that traditional drugs only target 10 to 15% of these proteins, and the remaining 85 to 90% of these proteomes are considered undruggable. And many of the cancer-driving proteins are unfortunately associated and belong in this category of undruggable proteins. I think before we kind of dive into a comparison between druggable and undruggable proteins, we really need to understand what a traditional drug actually does in the body and why that relates to proteins. A drug has a protein target, and when it meets a protein target, it initiates a binding process, and that leads to a therapeutic effect in the body. We see this being the case with many of the drugs that we use in just daily life. We see this with aspirin, which is targeting the COX enzyme. We see this with imatinib, which targets the BCR-ABL kinase. We see this with oseltamivir, which targets the neuraminidase enzyme. And so they all generally follow a pattern of recognition followed by binding, and this is only possible if they do have a sufficient residence time, which is the duration with which the drug stays binded with the protein target. Right, so what actually makes a protein druggable then? Examples of druggable proteins include things like kinases, GPCRs, other enzymes, and nuclear receptors. And the characteristics that make these types of proteins druggable is having a deep pocket, having a defined geometry, and also having favorable electrostatic interactions, as well as being very accessible to solvents. But many of these protein molecules are very tiny. They're typically under 500 Da, and they need a cavity for stable interactions. And that, I think, relates to why enzymes actually became the first drug targets. To kind of go over what an enzyme is, you probably know, but they're just biological catalysts that accelerate chemical reactions by lowering the activation energy. And why these enzymes are so easy to drug is because they need to bind to substrates. That's their main function, because they accelerate chemical reactions by lowering the activation energy, and they do so by binding to these substrates, right? And so they have very deep active sites from the get-go. They have highly conserved residues, they have very defined 3D geometries, and they have strong electrostatic interactions. And so when we put in a drug, that follows a competitive inhibition sort of pathway, wherein in a normal state, we just have the substrate and the enzyme, and that produces the product, accelerates the process of producing it. But when we have the drug present, they follow a competitive inhibition pathway, and so the drug occupies the active site that the substrate normally would have occupied, and the substrate is excluded from this sort of reaction, and the reaction itself is very inhibited. Right, so the rise of the undruggable proteins. Why did we get proteins that are undruggable? What changed is that as the genomic sequencing kind of process improved during the 1990s and the 2000s, researchers identified thousands of disease-associated proteins. And surprisingly, many of them were not enzymes. Enzymes which were very easy drug targets. Many of the proteins that were identified here were not enzymes. They were rather transcription factors, scaffolding proteins, adapter proteins, regulatory proteins, structural proteins, anything but enzymes. And the problem is that these proteins oftentimes lack the catalytic activities, and they possess very shallow surfaces, and they exhibit a conformational flexibility, and they function through for protein to protein interactions. A key classic example of this is the MYC gene family. So why can't we just design better drugs then in order to target these proteins that tend to exhibit less catalytic abilities or less defined geometry? Well, the issue is that many of these disease-associated proteins function by a protein-to-protein interaction. And many PPIs tend to involve large interaction surfaces, and they tend to be ranging from 1,000 to 3,000 A's, and these are stabilized by weak hydrophobic, electrostatic, and hydrogen bond interactions. But the problem is that traditional small molecule drugs are optimized to fit into deep binding pockets, meaning that they're too small to disrupt these extensive interfaces that are shown in protein-to-protein interactions. Take the MYC gene family, for example. They're considered undruggable because of the absence of stable binding pockets in their intrinsic disorder. So the consequence of having such a large undruggable proteome is that we have been able to identify thousands of disease-associated proteins, but many of these key disease regulators are these undruggable proteomes, and they tend to actually occupy a central position in downstream pathways, meaning that tackling their dysfunction is very, very important to being able to tackle the certain disease. So because we weren't able to tackle these important proteins because they're undruggable, a lot of our goals in drug targeting and therapeutics have changed from inhibition to elimination. So what does this mean? Traditional drug discovery methods were focused on the drug binding to the target protein, as I mentioned earlier, and the protein activity is inhibited, but this is only limited to druggable proteins, which is actually a big problem because undruggables constitute a large proportion of current diseases, right? But the limitation is that it requires a suitable binding pocket, and they're ineffective against most of undruggable proteomes. So the new strategy that's starting to emerge, or researchers are trying to turn an eye to, is the targeted protein degradation pathway, which is, instead of inhibiting the protein, we're removing it entirely from the cell. And this is a method that exploits the cell's quality control, or QC mechanism, and this involves pathways like the UPS, for instance. And that's something that we're going to be talking about in our next lecture.